protists, and then we'll start on fungi today as well. All right, so I want to re-emphasize these different life cycles because you're going to see this again when it comes to plants, and you're going to see it again when it comes to fungi. So, not all organisms exist in this world as diploid organisms, all right? There are organisms that exist as haploid organisms, where they only have one copy of every chromosome, all right? We are diploid. We cannot exist as haploid organisms, but that doesn't mean other organisms can't. So there are protists, in this case it looks like dinoflagellates, that exist primarily in the haploid form. All right? We're going to see this again in plants. Plants are weird because they can be haploid, diploid, they can be triploid, they can be hexaploid. Plants are all over the place when it comes to the number of chromosomes. All right? But in terms of protists, it's basically haploid or diploid. So one group basically maintain themselves in a haploid form unless something happens in the environment that triggers sexual reproduction. Actually, it's not even sexual, they call it sexual reproduction. It's not really sexual reproduction, okay? That's really not right. What it is two individuals fusing together, fusing their nuclei together to create a diploid cell that then undergoes mitosis and, meiosis and produces a bunch of haploid cells. All right? So, again, primarily haploid, environmental trigger causes the cells to fuse their nuclei, create haploid offspring. Okay. Then there are those that exist primarily in the diploid form. For example, diatoms. Diatoms are a little different in that the trigger for them to go from diploid to producing gametes, so mostly they reproduce asexually. All right, again. That was wrong. Again, asexually, they're just cloning themselves here, all right? Same thing here, they're cloning themselves. However, the problem with diatoms is that this top part here is smaller than the bottom part, so if you fit another piece in it, it's going to be smaller. It's going to get smaller and smaller until it reaches a minimal size. Once it reaches that minimal size, it undergoes meiosis, produces haploid gametes, all right? Not necessarily egg cell or a sperm cell, just haploid cells. These cells fuse their nuclei, produce a diploid cell that then undergoes mitosis and forms a mature adult cell, which then undergoes cloning, essentially. All right? So, again, asexual reproduction is dominating the primary form of reproduction here, but it will alternate with sexual reproduction when the size gets too small. Okay, then we have alternation of generations, and we can see various versions of this. And again, pay close attention because we're going to see exactly the same thing in plants. All right, so we have a diploid form of the organism. We refer to that as the sporophyte because that's the spore-forming form. All right, so in this case, the organism has a specific structure that undergoes meiosis. This, after meiosis, it produces these haploid spores. All right. Each spore then undergoes mitosis and expands in size and grows to a completely separate, independent, functioning organism. And in this case, the haploid form and the diploid form are virtually indistinguishable from each other. So you don't know what you're looking at. You don't know if you're looking at a haploid version of this brown algae or you're looking at a diploid version. Each haploid version now, it undergoes mitosis and produces gametes, sperm cells and egg cells. Now you have specific egg cells and sperm cells. They fuse their nuclei, you get a diploid cell, undergoes mitosis, and then produces the diploid sporophyte. So we have the gametophytes, the gamete-producing forms, and the sporophytes, the spore-forming forms. And that phyte, P-H-Y-T-E, indicates that we actually initially thought these were plants. All right, that's what phytos. Phyton means plant. Okay. Now, another version of alternation of generation is what we see here, that also brown algae, this is kelp in this case. Again, we have a primary diploid structure, and we have haploid structures, but they're very different in structure compared to each other. All right, so diploid is the major form. It has specific structures that undergo meiosis, produce haploid spores. Those spores grow out into individual organisms, but they tend to be very small. In some cases, you may not even really see them. All right? But they then produce sperm cells and egg cells. The sperm and egg cell combine, produce the diploid cell that undergoes mitosis and eventually produces the diploid sporophyte. All right? So those are the four forms of reproduction that we see in protists. So it can be sexual egg reproduction, can be asexual reproduction, can be cloning, can be alternation of generations, can involve egg cells and sperm cells or not. All right? Okay. So that's where I left off. So moving forward, protists play an important role both in a negative sense and a positive sense. On the negative side, they're responsible for a variety of diseases. So the Apicocomplexa, that's where we see malaria. Malaria is an international issue, even here in America. Now in the U.S., we now have cases of malaria, and we don't have a complete treatment for that. All right. So the best protection against malaria is to actually not get bitten by a particular genus of mosquitoes. These mosquitoes come out at night, and so the most effective form of protection is actually with netting around the bed that prevents the mosquitoes from getting to you. All right, we talked about dinoflagellates. These are aquatic protists that create red tides. What happens is shellfish are filter feeders. They filter these dinoflagellates out of the water. They accumulate them in their body as food. But the problem is that these protists contain neurotoxins, and so the more they consume, the more neurotoxin they build up inside the shellfish. And so then if we eat those shellfish, and we eat a bunch of shellfish, what we've done is what's called bioaccumulation, or what happens then is something called bioaccumulation, where each protist, each dinoflagellate, the amount of neurotoxin in it is not going to affect you in any way if you just eat one. But because those shellfish are concentrating those dinoflagellates in a single organism, you get lots of neurotoxin. All right, and then if you eat a bunch of shellfish, you get even more, to the point where it could actually become a serious issue. You could, in fact, get poisoned with a neurotoxin. All right, Giardia causes intestinal distress, diarrhea, Trichomonas I mentioned, sexually transmitted disease, Disca cristata, leishmania, causes skin sores, also affects your spleen and liver. Trypanosoma, that's sleeping sickness, primarily in Africa, can be fatal, and it's transmitted through flies. A different kind of trypanosoma is a big killer in South and Central America, causes Chagas disease. Then we have amoebic dysentery. We talked about the potato virus that wiped out potato crops in Ireland and caused a massive wave of migration to the United States. And then these microsporidians. Now, funny thing, I'm going to talk about microsporidians when I talk about fungi. So back in the day, microsporidians were considered protists. They're now actually considered fungi, and I just realized that that's still on there. And they cause, they affect the nervous system, they cause infections, they can be issues, but they're now actually classified as fungi. So, really should scratch that one off the list. Okay, malaria, just a little bit more about malaria. About 1 million people a year die from it. It's transmitted by the Anopheles species of mosquitoes. What it does is it attacks the red blood cells and destroys them, and if your red blood cells are destroyed, your body doesn't function properly, right? You don't get enough oxygen. So the oldest known cure was an extract from the bark of the cinchona tree. This tree grew in Venezuela, Colombia, so the northern part of South America. Story goes that there was malaria in Europe at one point, particularly in Spain and Italy. And back in the day, it was the Queen of Spain who basically ruled over pretty much most of Europe. And she got malaria. Now, she was lucky in that there were missionary priests who had gone over to South America, and one came back and had heard about this extract. It's very bitter, by the way. And so he treated her with it. She basically was cured, and she was so grateful that she then essentially sponsored men, to go back to South America and harvest these trees, get the bark, and extract the quinine, all right, to protect against malaria. So if you ever heard of bitter lemon, drink that bitter lemon. So the Brits, when they were in India, they drank bitter lemon. It was quinine spiked drink. All right. But the problem is that malaria has now basically become resistant to quinine, and so it's no longer really effective as a treatment against malaria. So malaria evolves like all other organisms, and so there's a constant chase to find something that will actually kill off the malaria causing organism, the protist, the Plasmodium. More recently, people discovered that an extract from a plant called Artemisia actually grows here on campus. It has a compound called artemisinin that apparently is effective against malaria, but I have a suspicion that since this was posted, that malaria has actually become resistant to it, so they would probably have to move on to something else. Talked about this before. Sickle cell anemia is the only known adaptation to malaria. If you have one allele for sickle cell anemia, you are protected against malaria. If you have both alleles, you get sickle cell anemia and you die. If you don't have either allele, no alleles, then you can get malaria and you die. But if you have one allele, you don't get malaria, you don't die. So,
Do you think it's ever possible to get rid of malaria?
is it what?
Do you think it's ever possible to terminate malaria?
To get rid of it?
Yeah,
I doubt it.
I would assume so too.
There's probably enough malarial organisms out there that should never get rid
Sure.
of. Okay. Harmful algal blooms. We used to refer to this primarily as red tide. That's mostly how we used to think about it. I mentioned red tide before. It's the dinoflagellates. They increase in numbers tremendously. I was just talking about it earlier. They get consumed by a shellfish. We consume the shellfish and we get poison. But that has now expanded into a number of protists, aquatic protists, that cause a variety of symptoms in people and animals. So you may recall, was it 10 years ago maybe already, there was a harmful algal bloom outbreak in Lake Opatcong, and you weren't allowed to go swimming there anymore. I don't know if any of you remember that, but it's not just something that happens in the ocean, it also happens in fresh water. It's becoming more and more an issue. And the problem is that these organisms thrive where there is lots of nitrogen in the water. And nitrogen comes from people fertilizing their yards, their grass, and then it runs off into the river brings it into the lake, and that nitrogen accumulates, and these organisms grow in huge numbers. And once they get beyond a certain concentration, they actually become toxic to us. All right. So normally in their low concentration, you can swim in the water; it's not an issue. But when they're in high concentrations, they actually cause a problem. And so we're seeing more and more of these harmful algal blooms, different organisms, dinoflagellates, but also diatoms that produce domoic acid. This causes loss of memory. Ciguatoxin is also associated with this. But what's also an issue with harmful algal blooms, so here's a picture of a red tide. You're also seeing this, where you get extremely high densities of algae, of brown algae, green algae in the water. And when that happens, it's not necessarily directly toxic to us, but what happens is, because there's such a super high density of these organisms, the top organisms block the light from the organisms lower down. And what happens is you get a massive die-off of algae. And those dead algae then get consumed by bacteria in the water, and those organisms consume oxygen in the water. So what you end up with is a large area of the ocean or the coast where the oxygen concentration in the water is extremely low. And when that happens, then all the fish in that area, anything that needs oxygen to survive, dies off. You get massive die-off of fish and other aquatic organisms, all right? And so you get what are called dead zones, right? So every year in the Gulf of Mexico, there's a massive dead zone that forms where the Mississippi River spills out into the Gulf of Mexico because the Mississippi River is loaded with nitrogen from runoff of fertilizer and animal feedlots and such. And then you get these massive algal die-offs and get these dead zones. Chesapeake Bay every year experiences this as well. And so this is a combination of two things. One is a combination of increased temperatures, because temperatures drive this as well. So the hotter it gets, the more this is a problem. And it's an issue of increased runoff of nitrogen and use of fertilizers. So we call these brown tides then. All right. Now, the news isn't all bad. We also depend very much on protists. For example, this organism here, Chromatococcus,
been discovered that that part of it is used to make a drug called astaxanthin. Astaxanthin is a strong antioxidant, so it's used for stroke victims, it's used to lower cholesterol. Interestingly, this organism is also fed to fish. So a lot of our fish these days is no longer caught wild, it's actually farmed in the ocean, primarily salmon. Most of the salmon that's available these days is actually farmed. The problem with farming it is that these fish aren't getting their natural diet. And so one of the side effects of that is that the flesh of the salmon in particular has a pale color to it, which is not what you normally expect to see. And so people are turned off by that. So to improve the coloring of the fish, they feed this Haematococcus to the salmon, and it gives them a red coloring that colors their meat red, which is what they would have normally been eating to get that red color as well. So it's not an unnatural thing, but it's ironic that it has to be added in. And then a big role that protists play, aquatic protists in particular, is that they are the bottom of the food web. All right? So everything else depends on the abundance of these photosynthetic protists, because they get consumed by heterotrophic organisms, which get consumed by bigger. All right? So if you affect this part of the food web, you affect everything above it as well. All right? We talked about this. We talked about the transfer of energy. So these are the primary producers here. So they're very important for maintaining healthy ocean aquatic ecosystems. And we depend on those aquatic ecosystems for our food and etc. Okay, any questions? Yes?
The alteration of generation,
Yeah.
that's going to be on our test on Thursday as well, correct? It's on the study guide, so I just want to double check.
No, should not be.
Okay,
No,
thank
should
you.
not be. That's a holdover, I forgot. Okay, no, that should not be. That will be on the next
Okay.
exam. And I'm pretty sure there will be a question about alternation of generations on the third exam. So you heard it here first. Any other questions? Are we awake?
Yes.
Okay, good. Glad to hear it. All right, so those were the protists. Now we're moving to the fungi, or the fungi. Okay, fungi are heterotrophs, exclusively heterotrophic. So protists can be heterotrophic or autotrophic, that is, photosynthetic or not. Fungi are exclusively heterotrophic, all right? And they have this ability to digest cellulose and lignin. And this makes them very unique among all the organisms out there, because pretty much those are the only ones that can do that, really. So they're extremely important for recycling organic material, making that carbon that's tied up in that lignin available again for other organisms. All right? So they're major decomposers. All right. Basic characteristics. All right? They have cell walls. Their cell walls have a unique composition. It's a combination of chitins and glucans. So it's a chitin-glucan complex. All right? So you're not going to find this in any other organism.
Insects have chitin, plants have glucose, some other organisms have glucans, but they have this combined here. All right, they have a cell membrane, they have organelles, but that membrane is not complete between cells. In other words, the cytoplasm of one cell is connected to the cytoplasm of the adjacent cell. All right? And that also happens because the cell walls are not complete either. All right? I don't know where it says that, but there are basically there are gaps in the cell wall between one cell and the next. So as a result, the cytoplasm is continuous from one cell to the next. All right? Now, another unique feature is that they're often multinuclear. So one cell will have more than one nucleus in it. All right? They reproduce primarily via spores. They live primarily as hyphae, so as filaments. And then there are a couple that exist as single cells. They occasionally form fruiting bodies. So when we think of fungi, we really only think of a fraction of the organism itself. When we think of fungi, we think of mushrooms, all right? But a mushroom is the fruiting body. It's the spore-producing structure of that organism. The actual organism, the bulk of that organism, is living in the soil primarily, or in the decaying wood, or maybe in a decaying animal. And so, over the last 20, 30 years or so, we've come to understand that some of these fungi, their hyphae, their filaments, can extend over a huge area. So they found basically one mushroom, one type of fungus that covered pretty much the eastern half of the state of Washington, which is thousands of square miles. All right, so they might actually be the largest organism on Earth, not by mass, but by extent. Okay, so as I said, they primarily exist in this filamentous form, and then there are some that live as single cells. All right? So a little bit more about that filamentous form. Those filaments are called hyphae. All right? Those hyphae primarily live in the soil or in a decaying organism. When they form the fruiting body, these hyphae condense, and then they form spore-forming structures. And here you see what I'm talking about. Here's the cytoplasm in one cell, there's the cytoplasm in the next. There's the cell wall, but there's a gap right there where the cytoplasm is connected from one cell to the next. And that's really important because, as it turns out, I'll come back to this later, the hyphae of these fungi that are in the soil, they connect to the roots of trees. And they act as essentially communication lines between the trees. So materials and signals move within these hyphae from one end of the organism to the other. All right? Okay. The ancestor of fungi is thought to be a protist. All right? So remember I just showed you that microsporidians were considered protists. That's what we're seeing here. They're now actually included in the fungi kingdom. All right? All right. Had I given this lecture, had you had this lecture last year, I would have told you there's 6 groups of fungi. In the meantime, now 2 of them have been split out, so now we have 9 groups. I think that's what it is. 1, 2, 3, 4, 5, 6, 8 groups. 8 groups of fungi. All right? We have the microsporidians. All right, so again, these are now fungi, no longer protists. They reproduce through spores only, they're parasitic, and they're unicellular. All right. Then there are the blastocladiomycetes, the neocalamasticomycetes, and the chytridiomycetes. These two here, the blastocladiomycetes and the neocalamasticomycetes, that's a mouthful. They actually were originally part of the chytridiomycete group, but in more recent years they've been split out. All right, and this is something you'll learn if you pay attention to taxonomy in organisms. There are what we call the splitters and the lumpers. So there are people who say, oh no, these are all very common. We put them in one group, we classify them as one kind of organism. And then there are others that will come and say, no. They really are different. We need to split them out as separate organisms. And sometimes one group wins and sometimes another group wins. And so taxonomy is actually often shifting over time. Anyway, so we have the blastocladiomycota. Their spores have flagella. So these three here are aquatic. Okay? So having flagellated spores makes sense. Their spores can swim through the water. They also have resting spores that can sit in the sediment for a really long time until conditions are favorable. They do produce gametes. They're saprotrophic, which means they feed on dead plant material. Okay, and they're parasitic, just like the microsporidians. The neocalamasticomycota. Every time I say that, I feel like is a supercalifragilisticexpialidocious. They also have flagellated spores. They produce gametes as well. They're parasitic, and interestingly, they have no mitochondria. Okay. Then we have the chytridiomycota, aquatic with flagellated spores and gametes. Their gametes also have flagella. The glomeromycota, they used to be part of the zygomycota. They got split off. They reproduce through spores only, and they are the primary group of what we call arbuscular mycorrhizae. AM, arbuscular mycorrhizae. Come back to this. Zygomycota, all right? Here the mycelia from two different organisms will touch each other. If they recognize each other as being from separate organisms, they will form a zygote, all right, a structure in which they fuse their cells, but they don't fuse their nuclei. All right, and they only reproduce through spores. Then we have the ascomycetes. These last three only reproduce through spores. Ascomycetes can be multi or unicellular. Basidiomycetes are multicellular. Ascomycetes form a specific structure in which those spores are formed. In basidiomycetes, they have pedestals on which those spores are formed. We go back to this in more detail and show you what I mean by that. Okay, so these are microsporidians. Again, they were initially considered parasitic protists. They're now included in the fungal family. They have an outer cell wall and an inner wall. They have this coiled filament that is spring-loaded. And so when this actually comes up against a cell that it can invade, because this is parasitic, it actually shoots this structure into the cell that it's parasitizing, and that then is how it extracts the nutrients from its host. So it's like a harpoon. All right? They lack mitochondria. They have chitin in their endospore wall. They produce those. All right. They're quite small. That's why the micro part. All right? The chytridiomycetes, that group of three, all right? They are aquatic, they have flagellated gametes, and their spores have flagella as well. So they undergo an alternation. Oh, hey, here we go. Alternation of generations, okay? So like I just showed you in the protists, we see this now in the chytridiomycetes as well. They form a haploid structure that produces gametes. One is bigger than the other, so we tend to assign that the female gender, and the smaller one we assign the male gender. But really, they're not male or female. They are just, one is bigger and one is smaller. Okay? They fuse their cells initially, and then they fuse their nuclei as well. Then they undergo mitosis. They form the sporophyte, essentially the spore-forming structure, which undergoes meiosis and produces the haploid spores that then produce the gamete-forming form. Right. There's a chytridiomycete on a pine pollen grain. Then we have the glomeromycetes. They form, so like I said, they're vascular mycorrhizae, and they only reproduce through spores. They form these spheres within which they form the spores. Now, I used to have a graph here that showed them with alternation of generations, but I was doing some more research yesterday, and apparently now folks have decided they don't undergo alternation of generations, so check that out. Okay. Zygomycetes, all right? There's that zygote that I'm talking about in a little more detail. You have, now these are land-dwelling organisms. They grow on cow feces, horse feces, things like that. Things with high organic material in them, lots of nutrients. They have their hyphae. Their hyphae will, if they come from two separate organisms, they will come in contact, they'll fuse, and then what they'll form is the zygosporangium, where the nuclei do not fuse. All right, this is another unique feature of fungi. In a lot of fungi, when you get fusion of cells, you do not get fusion of nuclei. All right? So you get this structure with separate nuclei in it. So it's diploid in the sense that there's two different nuclei in there, but they're not fused. They're separate. At some point, though, they do fuse. Those nuclei do fuse with each other, and then it does become completely diploid. And then those nuclei undergo meiosis, again form haploid cells, which then get put into a structure and form a spore, which then will grow into the next structure. All right. This group includes a group of fungi referred to as Pilobolus. Pilobolus means hat thrower. All right, these are the hats, and these are really interesting fungi because that part of the fungus is translucent and it's filled with water. And that's area right there, so these are spores. These are a whole cluster of spores here. And right underneath them, there's a weak spot here. And so when the sun hits, this water heats up. When water heats up, it expands. It exerts pressure, and enough pressure builds up, it actually shoots that spore cap off. And it can shoot it 10 feet through the air. All right? That hence the name hat thrower. we have the basidiomycetes. They form the spores on pedestals, basidia, bases, all right? So the spores are produced external on these basidia. Same thing, it's, as you see here, the spores land on the soil. They have different genetic makeup. If two different hyphae from two separate organisms come in contact, they will fuse their cells but not their nuclei, and they will form a multinucleate structure. That's typically what you see when you see a mushroom, right? Your basic mushroom. It's a multinucleate structure. It will then, on the gills, on the outside of the gills, it will form these basidia, and within them, then you get the fusion of the nuclei, making it diploid. All right, those nuclei undergo meiosis, form 4 haploid spores, which then get released and fall on the ground, and the whole cycle starts again. All right, so there's your basic basidiomycete. Here's another one. You may have seen this. There were examples of this. The geaster, the earth star. These are a group of fungi referred to as puffballs. They form the spores in the inside of this structure. When rain hits it, it compresses it. That increases the air pressure here and pushes the air out. And when the air gets pushed out, the spores get pushed out as well. Okay, ascomycetes are similar to basidiomycetes pretty much, except that their spores are formed within sacs inside of asci. And it's the same thing. Initially, the nuclei are separate, they fuse, undergo meiosis, form new haploid cells that get packaged into spores, and then those spores are released from these various asci. All right? Same thing, you have two separate hyphae coming in contact with each other, except now instead of fusing their cells, they actually create these external structures that then fuse. Again, only the cells are fusing, not the nuclei. And that forms then the fruiting body, the spore-producing structure. Here's an example of a ascomycete that looks like a morel. Very tasty. These are also ascomycetes, that green fuzz that forms on oranges when you leave them around for too long. Those are also ascomycetes. That is the source of penicillin.
Are truffles in that group as well?
I don't know if truffles are basidio or ascomycetes. They're one of the other. So it turns out that some ascomycetes, some basidiomycetes look very similar to each other. They're fruiting structures, they're spore-forming structures. So I don't. Okay. So as I said earlier, one of the unique features of fungi is that they excrete enzymes that can break down lignin. All right? So they have these fungal mycelia, these filaments, have a very high surface area to volume ratio. Right? They're these thin filaments that are spreading throughout the soil. And so that's why they have to be in a moist environment. They have to be in the soil because otherwise they would dry out pretty much instantaneously. All right? But they are really good at decomposing wood and particularly digesting lignin. All right? And lignin is just like it's just dense in cellulose. And cellulose is very dense in glucose. So lignin is like very dense glucose, which we know is an important component for cellular respiration. All right, so plants need carbon to undergo photosynthesis. They need it in the form of CO2. All right, they take in CO2 from the atmosphere. They put that into lignin, among other things, the plant structure. When those plant structures die and they fall on the ground, fungi will attack them, will decompose them, and as a result, they'll make that glucose available to other organisms. Those organisms and the fungi themselves use it. So the fungi use it, other organisms use it, and when they're respiring, when they're undergoing cellular respiration, they are releasing CO2 back into the atmosphere. And that then becomes available again. So fungi help recycle CO2 back to the atmosphere for plants to use, but they also make carbon available for other organisms to consume and function. All right? Now, probably fungi existed on land, not far out, basically right where water and land came together. There's this basic idea now that fungi helped plants move onto land. Okay, but they were not in high abundance until about 350 to, well, 300,000, 300 million years ago, excuse me. When we had what's called the Carboniferous period. This is a period of time on Earth when there were high CO2 concentrations in the atmosphere, it was warm, it was humid, and plants were growing like crazy. Massive amount of growth, which produced a massive amount of dead plant material on the ground. And at that time, initially, there weren't abundant fungal species, and so that plant material was just sitting there, it wasn't decomposing. All right? Which is why we have coal, because this is when coal got formed, when these massive amounts of dead plant material got covered up, got put under pressure, and transformed into coal. But it provided a tremendous opportunity for fungi to evolve because of this massive food source that was just sitting there waiting for an organism to take advantage of it. Fungi are the ones that evolved that ability to digest that lignin, and when that happened, their numbers went up like crazy. There's just millions of fossils of fungi that happened because they had this massive food source available to them. Nobody else was consuming it. It was out there, and so they could take advantage. Of course, once they decomposed all that wood, their numbers crashed again because basically they ate they ate all the food. And then the amount that was available was at much lower concentrations. Okay. This is also when fungi started, or actually before this even, fungi were undergoing these mutualistic relationships with plants referred to as mycorrhizae. Again, myco and rhizae. Myco refers to fungus, rhizae refers to roots. All right, there are two kinds of mycorrhizae. There are ectomycorrhizae and endomycorrhizae, or arbuscular mycorrhizae, the other term. Ectomycorrhizae, the hyphae, the filaments of the fungus, surround the root tip, and they do go into the they move into the root tip, but they do not move into the cells of the root tip. They go between the cells. That's why they're referred to as ecto, on the surface. Okay, so they'll go between the cells, but not inside the cell. That's what it looks like under a microscope. All right, then the other group are what we refer to as arbuscular mycorrhizae. All right, now the ectomycorrhizae are more common in colder areas, helping plants take up phosphorus. Ectomycorrhizae are important for helping plants take up nitrogen. Okay? Now, arbuscular mycorrhizae, or as we used to call them, endomycorrhizae, are referred to as such because their hyphae actually go into the cells of the root. So they're not that they go between, but they also go inside the cells of the root. Right? So they are mutualistic, but they are more aggressive in acquiring their nutrients from the plant root. They're acquiring them more directly. Okay, I mentioned this earlier. These mycorrhizae are not just around the root tips of one plant or tree. These mycorrhizae extend way out into the soil. This is how plants essentially extend their roots into the soil. They use the mycorrhizae to help bring nutrients in. But those mycorrhizae, as they travel through the soil and they encounter the roots of another plant of the same species, they'll connect up to those roots as well, and they'll form a connection between one tree and the next. This was discovered about 30 years ago or so by a woman in British Columbia, a professor in British Columbia, and she referred to it as the wood wide web. So the whole forest, if you go into a forest, the trees are actually really kind of one organism. They're all connected to each other. And if one tree is stressed, the other trees can move resources, nutrients from them to that stressed tree. So they act like a community of trees. They're not independent. They actually will support each other. People have measured movement of signals across these mycorrhizae is actually quite fast. And we know that the more mycorrhizae they are, the more plant species, the more greater the diversity of plant species there are, the more they grow as well. The bigger they grow. So mycorrhizae are really important. About 96, 97% or so of all land plants have mycorrhizae, have this fungal symbiosis. Okay, I also mentioned this before when I was talking about plant defense, defense basically. There are fungi that live in the leaves of plants that actually boost their immune system, so make them resistant to pathogenic fungi. All right? And then there are lichens. Lichens are a symbiotic relationship between a photosynthetic protist and a fungus. And they are extremely important. So lichens are extremely important because, so this is kind of what they look like in a schematic way. But lichens are very important because they can live where there's basically no soil. They can live on bare rock, they can live on bare tree bark, you see them growing on bark, you see them growing on rocks. They are the first organisms that move into, so if you have a volcanic explosion and you get lava flow, what you have then is just mineral. So you don't even have soil. You don't have soil. All you have is just rock. All right? Now, if you wait long enough, that lava, that rock, will turn into soil. And the beginning of that process starts with lichens. Because lichens can grow there. They need they're photosynthetic. They can kind of generate their own food, so to speak. And because of the fungi, they excrete these acids that can break down the rock, start forming a little thin layer of soil. As they decompose themselves, they add organic material to that, so you get mineral rock and organic material mixing, and that forms then the initial basis for other plants to move in and start growing there. And then they add more organic material, they break down that rock even more, and eventually over time you get actual soil. which is essentially mineral rock material combined with organic material. All right, so there's a good example of lichens growing on rocks with different species or variants. Often they have different colors. One of the cool things that I encountered, I don't know if you can see, but it's kind of green here, and it's kind of reddish up here. If you look at the PowerPoint, you'll see it more clearly. You can't actually see that. I can see it, certainly. This is in the desert of Namibia. These are fungi and lichens that are form what's called a cryptogamic crust. So they combine with photosynthetic bacteria and fungi, and they form this thin layer. Because actually what you're seeing here is sand, and on top of the sand are these flat pebbles everywhere, and they're red colored. And on those pebbles in the sand, you have this thin layer of these photosynthetic bacteria and fungi growing. They are critical for that ecosystem because that's where carbon is made available, that's where nutrients are made available, and that's what allows other organisms to exist in this desert that is otherwise just desert. I had read, so a little backstory on this. I had read about cryptogamic crust. I had taught about cryptogamic crust, but I'd never actually seen them myself. Never actually even seen pictures of them. And I was driving through Namibia, through the desert there, and my wife was sitting next to me, and I'm looking out the window going, oh, is that? And stop and take a look. It's like, no, it's just rock. You know, get back in the car and drive along. And all of a sudden, I saw this, slammed on the brakes. Cryptogamic crust! They ran out of the truck, looked at it, verified it, got some pictures of it. My wife thought I was crazy. She's used to it. All right. Now, a lot of fungi are also parasitic. They are parasitic to plants. Plants and fungi have co-evolved. Fungi are probably the biggest pathogens that plants have to deal with. All right, but it's not just plants, it's also animals, although for the most part, we're not really affected by fungal pathogens. Athlete's foot, diaper rash, ringworm, these are fungal infections, but they're not generally major issues for us. But plants have definitely been affected. They've pretty much wiped out the American chestnut. That's a fungal disease that has pretty much killed off most of them. So there you go. There's your oranges rotting, there's your corn rotting. You know, so from an economic perspective, these are big problems. And people are trying to find ways to prevent these fungal pathogens from destroying crops, right? Now, we do have products. We have inorganic products that we can, fungicides that we can use. But the problem is they're not just toxic to the fungus, they're toxic to everything that lives in the soil, they're toxic to other organisms. They're overall, they're environmentally not good, all right? They're bad. So people are looking for alternatives. How do you want to grow your crops, you want to prevent them from being destroyed by some fungus, but you also want to keep the soil healthy, so you don't want to spray pesticides. So what do? Well, one approach that people are looking at is inoculating the plants with a non-pathogenic version of a pathogenic fungus. This is like vaccinating the plant. It's the same way that we get vaccinated. You get exposed to a non-pathogenic form of a pathogen, a virus, let's say. Your body builds up an immune response to that, so when it gets exposed to the actual pathogen, it can respond to it quickly. Plants can essentially do the same thing. You expose them to the benign version, the non-pathogenic version.
it doesn't kill the plant, right? You then, if you expose the plant to the pathogen, it pretty much kills it off. But if you pre-expose it to the non-pathogenic one and then expose it to the pathogen, in fact, most of the plants will survive. All right, so it essentially creates an immune response in the plant. Not that it's creating B cells or T cells. What it is doing is it's ramping up the plant's chemical responses to that pathogen, so it can fight it off. All right. Insects and animals are also affected by fungi. All right, so here, that's a fungus that's infected that insect there. A major issue is something called Dutch elm disease. It's not that the Dutch created this disease, it was first discovered in the Netherlands. This is a fungus that affects the growth of elm trees. What happens is when the fungus gets in the tree, the hyphae grow in the water, the xylem cells. And when they do that, they block the xylem from transporting water. And if the xylem isn't transporting water, the leaves die, and the leaves die, the tree dies. All right? It's not instantaneous, it takes a few years. Now, how does that fungus get into the tree? That's the interesting story. So there are beetles, these little beetles. This is what the emerald ash borer, heard of the emerald ash borer? It's killing off all the ash trees. All right? It's a little beetle. And these are referred to a group of beetles called bark beetles. What these beetles do is the females will chew their way into the bark, into the inner layer of the bark where there's live, nutritious cells. And they will eat up and down and around and create little tunnels, and they'll lay their eggs in there. And so when those eggs hatch and the larvae are growing, and that fungus is if they do this in a tree that has the fungus, those larvae pick up the fungus. They mature into mature beetles. When the beetles are mature, they fly out of the stem. Those are the holes. Those are the exit holes. They fly out and they go in search of another tree, another elm tree. But in the process, they're carrying the spores of that fungus. So when they drill into the tree again, they're introducing the spores of that fungus. And that's how it gets transmitted from tree to tree. So, again, American elm barely exists. There are very few trees left in the U.S. of American elm because of Dutch elm disease. All right? Another big issue. This is major. What time is it? A major issue facing amphibians right now is there's a massive die-off. All right? We're losing huge percentages of amphibians, frogs and salamanders. And one of the causes is this chytridiomycete, this fungus that latches onto the eggs and the larvae of these amphibians, frogs and salamanders. They don't necessarily kill the frogs and salamanders directly, but they can cause deformations. And so they don't function properly, they can't eat properly, and they die. They get stressed and they die. Now, this is not the only cause, but it is a contributing factor. Other factors are increased warming, increased pollution, increased ultraviolet radiation. All of these increase pesticides and stress. These increase the stress. As they become more stressed, they become more susceptible to this fungus. And so the fungus is just kind of like the final straw that breaks the camel's back. Okay? This is a fun one. The Ophiocordyceps. This is a fungus that shoots its spores out. They land on an ant. All right, it's mostly ants, but it can also be beetles. It can be praying mantises, etc. It lands on these insects.
insects, it gets under their outer skeleton, and then it starts expanding inside the body. What it does is it actually goes to the brain of the ant, and it takes over the brain function, it takes over the behavior of the ant. And it makes the ant climb to the top of a leaf or a plant stem, and then it'll just clamp its jaw in and die. And then once it dies, the fungus grows out of the ant, produces that spore-forming structure, releases the spores that then fall through the air down on the ground where the ants are running around, and it falls on the ants, and then those ants that get infected, again, basically become zombies and start crawling up. After a couple days, they crawl up to the top of the plant and clamp their jaw and die. This is referred to as the zombie fungus. Okay, again, all bad news, right? We rely on fungi for a number of things. First of all, for plant growth, mycorrhizae is extremely important, all right? But, hey, we get our alcohol from fungi as well, all right? Yeast. Yeast are fungal organisms. They're single-cell fungal organisms. When they are exposed to anaerobic conditions, they form ethanol. All right, if you remember back when we talked about cellular respiration, under anaerobic conditions, there's two possible pathways. One is ethanol production, the other is lactic acid production. Yeast produces ethanol. That's the alcohol that you get. All right, so we can have alcoholic drinks as a result of yeast fermentation from yeast. Cheeses, flavoring, blue cheese, for example, that's a fungus in the cheese. Bread, again, that's yeast. But in this case, we're keeping it aerobic. And the yeast are producing CO2. That's what makes the bread rise. It's the CO2 produced by the yeast. All right. Soy sauce is fermented soybeans. Tempeh has this. These are wheat kernels surrounded by this fungus that grows around them. It's a major source of protein. Penicillin, all right, we talked about that before. Penicillin was the first antibiotic that was discovered. It was discovered really by accident. Basically kind of results of not very clean lab practice. So Fleming, a scientist that discovered this, was a microbiologist. He was growing fungi on plates. But at the same time, apparently he had an orange or a couple oranges in the lab that were rotten, and they had this green fungus growing on it, this penicillin. You know who that was at the time. And some of the spores got on his plates. And when he came back after the weekend, he noticed that there were these dead zones in his fungal plates. And he tried to figure out why parts of his fungal growth had died off, particularly in these very concentric circles, right? And so he eventually figured out it was the spores from this fungus that were doing this. And then it was like, aha, hey, these spores are killing this fungus, maybe we can use that. So then they had to figure out what was the active compound. That became known as penicillin because it came from the penicillium fungus. All right? And then basically that then got developed by other people as a treatment for, as an antibiotic, not just for fungi, but for bacteria as well. Actually, he was growing bacteria, sorry, it wasn't very fungi, he was growing bacteria. I said fungi. He was growing bacteria in this place. So it was killing bacteria, and then he said, I'm killing bacteria. We could use this. But anybody know when that discovery took place? How long have antibiotics been around? Anybody know? All right, we'll do it. Okay. Antibiotics have been around for 10 years. Raise your hand. 20 years. 30 years. 40 years. 50 years. 60 years. 70 years. 80
Longer.
years? All right, they've been around since after the Second World War.
The hell?
Basically since the late 40s, early 50s is when antibiotics were first introduced in a medical use. So if you figure 1950, so 70 years roughly, 70, 80 years. When I was a kid back in the 60s, antibiotics were still pretty new. All right, penicillin was the only antibiotic. And in fact, it turned out that not everybody could get penicillin because some people are allergic to it. Now we have a huge variety of antibiotics, but that was the first one. It was the only one for a while. And so before that, if you got an infection, you actually had a pretty decent chance of dying from it. Whereas now, we don't even think about that. You cut your finger, you go, at least I go, whatever. Some people freak out.
Bless you.
For the most part. Actually, now the problem is we're overusing antibiotics. Yes,
All right, you answered the question.
I did.
Yeah.
What was my answer?
Oh, you said people were dying.
Yes, people were dying from, yeah. So, Civil War here in the U.S., most of the soldiers who died, and there were millions that died, they didn't die because they got a bullet through the heart or through the head. They died because they got wounded and they got infected. Most of those deaths were through infections, not through the actual killing them through the bullet. So, yeah, I mean, we're very lucky to have this, but now we actually have a different problem. Now we have pathogens that are resistant to our antibiotics, and we're losing the ability to treat a lot of infections because we've overused those antibiotics.
Well,
They evolve, they're organisms, they evolve. So, we actually need soap. Best thing you can do, don't use antibiotic soaps. Don't wash your hands with antibiotic soaps, just regular soap. That's all you need. Plus, it turns out, the more you actually expose yourself to bacteria and fungi around you, the healthier you actually are going to be. Yes?
Isn't that going to develop an intrinsic macrophages against like drug resistant?
Yes, okay, so because antibiotics aren't working so well, now the question is there some other treatment? And there are viruses that attack bacteria. They're called phages. Macrophage is something in your body that attacks, but phages they're phagocytic, whatever. They eat bacteria. Bacteriophages, that's what they're called. Sorry. So, they're just referred to as phages. So the question is, could you introduce a virus that specifically targets a very specific bacteria? Possible. Now, one issue is that there are bacteria on your skin that actually protect you, but they can also become pathogenic. So if you get rid of all those bacteria, you get rid of the pathogen, but you also then are no longer protected. All right? This is one of the major issues facing humanity right now, is antibiotic resistance, the overuse of antibiotics, and not understanding really the role that bacteria and fungi play in human health, in fact. Okay. I don't know. Boy, you guys aren't asking enough questions. I'm just shooting through this material like nobody's business. So that's it for fungi. I'm going to call it quits for today. I'll start something new. So that's it. See you on Monday.